In this video, we are going to examine the effect of chromosomal aberrations during meiosis and on the fitness of an individual. Say we have an individual 1, with the following chromosomes. One of the chromosomes has a normal gene order, and the other one has a section that is inverted along with a centromere called a pericentric inversion. If we were to make gametes with these chromosomes, we would have to contort them into a special shape called an inversion loop, so that the loci match up and crossing over can occur. To diagram this, we first draw the normal chromosome in an omega shape. Don't forget that these chromosomes are replicated, so there are two strands in our diagram. Next, we then draw a loop-de-loop shape inside the omega for the inverted chromosome. We then label the loci with all of the affected loci on the arc of the inversion loop. We then draw a straight line down to denote their location. Let's say we have a crossover in the J and K region. From this, what gametes will we receive? Let's start with the chromatids not involved in the cross. We'll make a check mark at this end of the non-inverted chromosome and trace it with a color until we reach the other end, where we leave another check mark. When we list the loci through which we pass, we get the gamete with a normal order: M, L, I, centromere, J, K, H, G. Next, we trace the unaffected chromatid of the inverted chromosome with another color, following the same procedure. Now, let's trace this end of the normal chromosome. When we reach the crossing region, the path crosses over onto the other chromosome, which changes the loci order. When we rewrite our gametes, the loci follow the order of the non-inverted chromosome until the crossover region, where we have a repeat of loci already in the chromatid in absence of some loci. If we trace the last available chromatid, we notice a similar pattern and receive this gamete that has duplicated and deleted genes. From this, these recombinant gametes are not viable, since they do not have all the necessary genes in the right quantities to create viable offspring. Let's compare this to another individual 2, with these chromosomes. These chromosomes are identical in their gene order, so there are no special crossover structures. Lining these chromosomes up, we have a crossover event occur between the J and K regions. After crossing over, we have two chromosomes that, when separated into chromatids, are all functional, since they have all the necessary genes to create viable offspring. So which organism has better fitness? The answer would be individual 2, because this individual has a greater number of viable gametes compared to individual 1, and thus has a greater chance of creating viable offspring.